So, 6.5, regulation of enzymes. So, metabolism, that's like, you could define metabolism as the sum of all the chemical reactions in the cell. So, you could think of a cell as just like a set of organized chemical reactions. And, sometimes the chemical reactions, or really most of the time, we want to control how fast or how slow they happen. In other words, you don't want to do all the chemical reactions as fast as you can for as long as you can, because then you would not survive. There are times when maybe we want to do more, if we go over we have like catabolic reactions, maybe we want to do a little more catabolism. We want to take more of your polymers, like right now, a lot of you just had lunch. You want to do catabolism, take your polymers, break them into monomers. Or maybe you want to do anabolism. So maybe you want to build something. Like you took those monomers from the food you just ate, and you want to build those up into some polymers, some like proteins or lipids, some hormones that you need to signal stuff to other cells or maintain the shape of your cells. Enzymes help us with those processes. Was catabolic exergonic or endergonic? Exergonic, right. Catabolic, that was exergonic. Remember, exergonic, that is the delta G there is negative. That means you release energy. If I draw the exergonic reaction diagram, G, that's a measure of free energy. So in that, the reactants start at a higher free energy. We then have that activation energy, and then the products start at a lower free energy. These are your reactants, those are your products. This right here, that's your delta G. So because we went from higher to lower, that's why it's negative. There's a release of energy. It's a delta G that's negative. Okay? So it's kind of trying to help you connect some dots here from earlier in chapter 6. Entropy. What does entropy look like for catabolic reactions? We increasing entropy or decreasing entropy? Entropy is disorder. If you do a catabolic reaction, are you increasing disorder or decreasing disorder? Well, increasing, yeah. So that would be an increase of entropy. Remember, it's a little bit backwards of what you might think. The universe wants disorder. So increasing entropy, increasing disorder, that's what we're doing catabolic reactions. That's favorable. That's another word to kind of know. Catabolic reactions are favorable. You know it's favorable when you have a negative delta G. You have lower free energy. Why is there because for G, we also call that potential energy. So in chemistry, sometimes you would see these diagrams, the x-axis is time, but sometimes you'd see the y-axis as potential energy. How would that be potential energy? What would that mean for this polymer to have potential energy? What are they saying? Or where is the potential energy stored? If you have glucose from the food you just ate, how exactly your food have a high G, a high free energy, high potential energy? Yeah, go ahead, Thais.
Is it the bonds
the bonds, yeah. The bonds that hold together your food, when you break that down, you're releasing that energy. This is the energy that gets released when you're breaking that polymer, like your glucose, into your monomers. Okay? The reason why this would increase entropy is, if you remember glucose, C6H12O6, it's one organized molecule, and then you're going to break that down into CO2 and H2O. And multiple CO2s and H2Os. You're breaking into more monomers, more pieces. That's why it's higher entropy. Okay? So then anabolism is more or less the opposite. So this would be monomers to polymers. Technically, it's not all, there's other examples of catabolic and anabolic reactions that are, but think this. That's a good starting spot for us. This would be endergonic. So endergonic, what that looks like for the reaction diagram is your reactants start at a lower free energy and your products end at a higher free energy. And that's why here the delta G is positive, equals positive. You raise the free energy, that potential energy of your products. That would be like you took the monomers, maybe the amino acids from your lunch, and you made a protein. That protein, the bonds in that protein store a bunch of energy that, you know, your body can use to like for structure or to like be an enzyme or something. Okay? Yeah, so delta G is positive here. And we call this not favorable or non-favorable. What that means is like, it's just because you're ending at a higher free energy, that's just, you got to put in a lot more energy. Now, remember, even if it's an exergonic reaction where you end at a lower spot, all reactions, we have to put in some amount of energy. And this is trying to get into like how enzymes help us. This is the amount of energy you have to put in to make those reactions happen. What is that called? Can you remember? That red, what I did in red there, what would you call that? Yeah, go for it, Lucas. Activation energy, yeah. So this would be your AE, your activation energy, okay? So even though I'm releasing energy here, I still have to put in, it's like an investment. You have to invest, you gotta invest money to make money, right? You gotta invest some energy to get out even more energy than what you invested. Even if it's an exergonic reaction, even though we're overall releasing energy, okay? Now, how do enzymes help us? Well, enzymes reduce the activation energy. So what an enzyme does is, an enzyme lowers how high the hill is. It lowers the activation energy, but you still end at the same spot. A very common misconception is you still end here at the same spot, meaning the delta G of the reaction is still the same even if we have an enzyme, right? All the enzyme did is lower that activation energy. Really important idea, okay? Cool, so that's just kind of pretty quick review of a lot of the other stuff in chapter 6. Now then, okay, enzymes help us speed up these chemical reactions. We want reactions to be turned on and turned off. How do we regulate these enzymes so you're not just like all gas, no brakes, you know, with your chemical reactions? Well, there's two main ways you can do it. One is you just turn on or you turn off the genes. Remember, genes, that's on your DNA is organized into genes. Genes have the information for one protein. Enzymes, generally speaking, are proteins. Sometimes they can be RNA molecules, but typically they're proteins. So if I want to make more of an enzyme, I just turn on that gene and make more protein. If I have enough enzyme, you turn off that gene. Pretty simple. Where it gets a little more complicated and kind of clever, and more of what we'll get into today, is how we can regulate the activities of enzymes that are already formed. So we already have enzymes made, but we can actually turn on and turn off already made enzymes. So this is more turning on and off genes. That's more like long-term regulation of enzymes. So you'll see this with like people who take like you have high cholesterol. You'll take a medicine like a cholesterol medicine that lowers your cholesterol, but over time that medicine doesn't work as well, and the doctor starts having to up the dose of that cholesterol medicine. Why? What's going on there? Any thoughts? Lucas, you go. Say it again.
The enzymes are like saturated, so like it works.
It wouldn't have to do with the saturation of the enzyme. I like what you're thinking. No, it wouldn't be so much that. Thinking about this point here, this first bullet point. Why would you have to keep upping the dose of that cholesterol? Basically, the cholesterol medicine is like taking an enzyme that helps like you regulate the cholesterol levels. Okay, so somebody who has high cholesterol, their body will naturally make proteins that can regulate their cholesterol. But if you start supplementing them with those proteins, with the medicine, their body will start turning off the genes to make the cholesterol because they're getting it from some other place. So then you have to begin upping kind of that dose of the cholesterol medication. So in other words, you're kind of fighting your genetics with like medicine. That can be a very common theme with medicine, is like trying to get the dose right because your body will like naturally adjust to like the outside source that you're getting. Anyways, then most of today again is like going over how do we regulate enzymes that are already made. Okay, let me get my cursor over here. So there is something called allosteric regulation. Allosteric regulation. Allosteric is referring to binding to somewhere that's not the active site. So like here, we would call this an allosteric site. Allosteric site. Because like here's your enzyme, and this is actually a complex of 4 enzymes. This is the active site where the substrate wants to bind. The allosteric site, it's a part on the enzyme, but it's not the main part of the enzyme that does the reaction. That's an allosteric site. Okay? So when they say allosteric regulation, we're talking about these things that it could be an activator or it could be an inhibitor, these molecules that can bind to the enzyme and either turn them on or turn them off. They bind to a site different from the active site. This was similar to non-competitive inhibition that you learned last week. What was competitive inhibition? Do you remember? Yeah, go ahead, Tina.
It binds to the active site.
Yeah. So you have competitive inhibition. You have like, here's me. My laser pointer is competing with this substrate. I beat this substrate to the active site, and now I'm occupying the active site, and this substrate can't get there. I outcompeted that substrate. Non-competitive inhibition is saying, instead of me like competing with you for the active site, I'm just gonna like get rid of the active site. It's like cheating. It's like when you play like rock, paper, scissors. You're in there with somebody and they're like, rock, paper, scissors, snake or something, they just like bomb, they just like change the game on you. That's kind of what non-competitive inhibition is. It's like this inhibitor just changed the game. It just like literally blocked the active site, so this substrate can't even get to the enzyme. Okay? Now right here, this is kind of interesting. So enzymes, when you learn about enzymes in like freshman bio, you kind of just see them as like this stationary molecule with an active site, but they actually will like oscillate between being active and non-active. Active, non-active. Kind of like how you might walk. You could run, you could make it more active, or you could just sit down and make it more non-active. The reason for that is, again, naturally we don't want enzymes always being active. So there's like a baseline level of activity that enzymes naturally have. If I need that enzyme to be more active, that's when you put in the activator that binds to the allosteric site. Or if you need the enzyme to be not as active, you don't want to do that metabolic pathway because you have limited energy for your cells, you'll then use an inhibitor to block that active site. Okay? It's very like elegantly simple. It's like I don't know that like once you see like that's how it works, it's like that actually makes a lot of sense that it would work that way. Notice here that there are 1, 2, 3, 4 purple monsters there. These are 4 different polypeptides. This is a protein. This would be a polypeptide. That'd be a polypeptide. Meaning some enzymes are a complex of multiple polypeptides. Some enzymes could just be this one enzyme, but whenever you do allosteric regulation, it's usually going to happen when you have a complex of enzymes. The reason for that is if you just had this one enzyme, you don't really have like there's less space for there to be an allosteric site. But if you have a complex of like proteins coming together, now you have these kind of random like almost dead spaces where you could have an allosteric site. Okay, kind of a random point, but it's technically a thing. Okay, so let me make sure I said everything here. See, that was all called allosteric regulation. Think of it's very similar to non-competitive inhibition. And it's either you inhibit with an inhibitor or you stimulate with an activator. They each are binding to that allosteric site. So they do that by binding to the allosteric site. That's how they inhibit or they stimulate. Yeah. So that's just describing how they're not going to the active site, they go to a different site. Yeah, that's it.
What's the difference between allosteric regulation and non-competitive?
Not really much of anything. I mean, non-competitive inhibition is more of like unintentional. Like it's like you could have a poison that could like shut off your enzymes by binding to like an allosteric site. Allosteric regulation, like regulations, like kind of implies like we are intentionally trying to control it. So like, but functionally they behave the same way. Like they're just binding to a different site from the active site. Okay. All right. This first bullet point, that's what I was trying to show you where I was numbering the 4 different purple monsters there. So usually allosteric regulation is going to happen when you have a complex of enzymes. Enzymes that are made of multiple polypeptides. If it was a smaller enzyme that was just one polypeptide, that would be more where you just kind of have to like turn it off or turn it off more just by like competitive inhibition, like just by having something that can shut off that active site. And again, the enzyme will, even without an activator or inhibitor, the enzyme will naturally flip between being active and inactive. Okay? We can just change that baseline rate by doing the activator or the inhibitor. And then how exactly the activator like will allow the active site to be open is it stabilizes the shape. So like basically like when it by binding here, it's kind of like when you have like it'd be like my fingers are like the activator, like my fingers are stabilizing this open shape of this this is a paper clip. What do you call this? A clip? This binder clip? I'm stabilizing, I'm like an activator stabilizing the shape of this. You know? I don't know. And then like the spring or like the natural shape of the metal is stabilizing the inactive shape here. Okay. All right. Now another kind of allosteric regulation is cooperativity. In Mastering Bio, they kind of have a trolley kind of question where you'll have cooperativity and allosteric regulation as answer choices. And something you got to get used to in AP classes is like the best answer questions. You know, so cooperativity is a specific kind of allosteric activation or regulation. So this is B. A was that last picture I showed you. So cooperativity is really interesting. So in cooperativity, if one substrate binds, if I can get, I have 4 different polypeptides here. If I can just get my substrate to bind to one of my active sites, that will then open the door for all the active sites. Think of it like a master switch with like electricity. I don't know if you ever like witnessed one of those. It's like you could have individual switches to turn on and off lights, or you could have a master switch that turns all of them off at once, or all of them on at once. So it's like it's an efficiency thing. You're trying to go to bed, it's nice if you can have one switch that turns off all of the lights. Or if you're, this is how we, the hemoglobin molecule in our body, this is how hemoglobin will work for carrying oxygen. Where if you can just get one oxygen molecule, let's say this is oxygen, you get one oxygen molecule to bind, you then open up the space for all the oxygen to bind. It's really efficient that way. And then the reverse will happen. So don't miss this. The reverse will happen here. If I lose one of these, so like if I need oxygen in my finger here, once that the hemoglobin and the red blood cell gets to my finger and it senses the pressure difference of oxygen, one of these oxygen molecules will leave the enzyme. Well, it's not an enzyme, it'll leave the molecule, and that will then trigger all the other ones to leave. It's almost like sheep. It's like a herd, like flock behavior, herd behavior. You know, it's like what one sheep does, the other ones just follow. Once this one substrate leaves, the other ones would leave too. Or when one binds, the other one binds. It's set up to where they all kind of follow each other. They're all cooperating with each other, right? There's nothing worse than like you're trying to like lead a bunch of sheep and then you get one that doesn't cooperate, right? That's why it's called cooperativity. All the other active sites are cooperating with that other active site. Okay? You good here? Make sure I said everything here. Yeah, you bind one, that will then it locks all the other ones in the active shape. So one binds, then all of them combined. Right? Just like kind of like a master switch. Like rather than having to like go and turn on all of them, you could just turn on one. Okay. And basically it's trying to say it's very efficient that way. So you can quickly turn on and turn off enzymes with that kind of mechanism there. Okay, I want to talk about a really, really important pathway or way that we regulate these metabolic pathways. Because I've just been kind of talking about enzymes and reactions as if they're these like separate things, but you got to remember that chemical reactions are almost like this pinball game where they're all connected to each other in a way. Or like an assembly line is a better way of thinking of it. Where if I jump to this picture, I'm starting here with threonine, and I want to turn threonine into isoleucine. To do that, I have to send this threonine molecule through 5 different enzymes to get to my final product. This is like an assembly line, right? So what can happen is, you know, if you're like you can imagine like an assembly line. If it goes too fast and you just keep having this like buildup of products, this would happen like where I taught middle school. We would order supplies and we literally had like 500 glue sticks. Because every year we would forget to remove the glue sticks and check the cabinets and make sure we had it. And so we just end up having this like oversupply of glue sticks. And I wish there was a way for the glue sticks to know once the drawer was overflowing that they could go in the computer and turn themselves off. Well, fortunately, our bodies do that. When you make the isoleucine, this isoleucine can actually feed back and turn off this first enzyme in the pathway. That way you're not over making things that you don't need. As I said, it's like you wouldn't maybe like think to do it that way, but once you see that it works that way, it's actually kind of cool and very like, oh, that makes a lot of sense, right? It's super efficient. A lot of metabolic pathways work this way. The end product will then block the first enzyme so you don't make more than you need. And then this is important. So this is here, it's blocking the active site. So this isoleucine it bonded to, that's an allosteric site. That's your allosteric site where it bonded. Now, okay, we've used up all the isoleucine in the cell. Oops. Well, now this isoleucine, the isoleucines that are blocking the enzyme in the first path, they're going to unbind and we'll use them up. But because they've left, now we know, oh, we need more. Now we're going to like open up that enzyme and we start the production process again. It would be like the glue sticks knowing that like, oh my gosh, we need to make more glue sticks. I don't know. Is that making sense? Makes sense to me. I think it's kind of cool. Anyways, remember this is allosteric because it's not competing, or it's non-competitive. It's not competing with the active site. It binds to a separate site from the active site. All right. Yeah, the last idea is another, it's a little less common way, but it's still an important way of how we can regulate these metabolic pathways is by how you organize them. It would be like a way I could control this classroom is by how I organize you, right? If I put all the, like, this happened like when I had like 7th graders. So I put all the boys together like in the back. That's not a very good way to organize a classroom, not have like chaos maybe, you know? Or like all of the super talkative people together, they just like could create chaos. So we could organize the enzymes so you could effectively control these metabolic pathways. Let me show you the mitochondria. This is how we might do in the mitochondria. Mitochondria, powerhouse of the cell. This is how we do cellular respiration. Some of the enzymes for cellular respiration we have in the matrix, or the matrix is like the cytoplasm of the mitochondria, in the empty, kind of the free space inside the mitochondria. other enzymes we have on the inner membrane of the mitochondria. Think of it like if you had multiple air conditioner units in a big building. Why would that be helpful? Why would it be helpful to have not just one giant air conditioner for like this whole high school, but if we had multiple smaller air conditioners? What's the benefit of that? Yeah, Libby, go ahead. Okay, let's imagine like the one big one could reach the air just as fast as all the smaller ones. You know, what could go wrong? I guess if you just had one big one versus many small ones. Yeah, go ahead.
The big one could break down and speed.
Yeah. If the one big one breaks, oops, all of the AC's out, right? Whereas if you separate them into smaller compartments, then if one breaks, you're not like breaking all of them. So in a similar way, like I don't know, like if by separating these enzymes out in different areas, it's kind of a way to like ensure that like if you have an issue over here, it doesn't like affect all the enzymes over here. Anyways, efficiency is also another area where if they're all working together, it's more efficient that you keep them in the same area. Like if you're doing a group project, it makes sense that all of you or all four of you stay at the same table and not that you're separated from different tables. That's not very efficient because it's hard to communicate with each other. So efficiency is another reason why like organization is pretty helpful. Okay, so looking back here, kind of a lesser detail, but like enzymes, like their main job is to speed up chemical reactions, but they also like are useful for like structural parts. Like they literally can like be like a column that keeps up like the mitochondria. It's almost similar to like in the cytoskeleton, how like you could have the cytoskeleton would be used for structure, but also it could be like a highway to transport things. So enzymes will also be structural parts of the cell. Yeah. Anyways, enzymes that are involved with like cellular respiration, you organize them in different compartments. It's a little more efficient that way. Okay, there we go.